That helps, and we will hopefully get something on the book that's going to have as few conflicts for people as possible. Okay, well, last time I got to this point in glycolysis where I was talking about that substrate-level phosphorylation that is involved in making ATP. So making ATP is a critical component of glycolysis. We remember that in the second phase of glycolysis, we basically have two of everything down here. So we have two G3Ps, we have two of those, we have two of those, we have two of those. So therefore, at this point, we've made two molecules of ATP. Now, two molecules of ATP are significant because we use two molecules of ATP to get the process started. So at this point in the process, we have broken even. We've completely broken even in the process. We've gone through one oxidation. We've made two ATPs after using two ATPs. Everything else in the process down to here is getting ready for the last reaction. And the last reaction is what I like to call the Big Bang. And we'll see why I call it the Big Bang in just a bit. Before we get to the Big Bang, though, I need to talk about this reaction, which is the 3-phosphoglycerate to 2-phosphoglycerate. And the enzyme that catalyzes it is called phosphoglyceromutase, or you can also call it phosphoglycerate mutase. Either of those are acceptable. And this is a very interesting and unusual enzyme. So phosphoglycerate mutase works differently than an isomerase. So you might say, well, this is just a simple isomerization. We're simply moving the phosphate from position 3 to position 2. But it's actually somewhat more complicated than that. So I want to tell you a little bit about what's up with that and why this is of significance. So a mutase works not by simply moving a part of a molecule to another place on a molecule. Instead, it makes an intermediate that has both features of the molecule. So for example, this mutase goes from 3 to 2 by putting a phosphate on position 2 to start. So it's got a phosphate on position 2, which is the final product. It's got a phosphate on position 1, which is the starting product. And that molecule you've heard of before. It's known as 2,3-BPG. 2,3-BPG was a molecule we talked about with respect to hemoglobin. And if you remember, hemoglobin, when it bound to 2,3-BPG, released oxygen. And I told you that 2,3-BPG was an indication of high metabolism. High metabolism. All right? Well, this intermediate 2,3-BPG that arises in this reaction, this intermediate can actually be released by the enzyme in small quantities. That is, a small percentage of the 2,3-BPG escapes from the enzyme before it becomes 2-phosphoglycerate. Most of the enzyme simply takes the phosphate off of position 3, and you're left behind with 2-phosphoglycerate. But a small percentage escapes as 2,3-BPG. The more of it escapes, the more of this process is going on. Right? So the more glycolysis is going on, the more reactions we will have, and therefore the more 2,3-BPG will be released. So 2,3-BPG turns out to be a good indicator of high metabolism. It arises only because of this stable intermediate caused by the mutase. I see some frowns. Is that making sense or not making sense? Everybody understands why 2,3-BPG is an indicator of high metabolism? You could tell me on an exam? Okay, okay. So 2,3-BPG is an intermediate because mutases make an intermediate that has properties of both the substrate and the product. In this case, the substrate, the only difference is the phosphate's on 3 versus the phosphate on 2, so the intermediate has a phosphate on both. The cell, phosphate, there's plenty of phosphate floating around the cell. Okay? So this intermediate, 2,3-BPG, is stable and can be released by the enzyme in small quantities. Let's say 95% of the 2,3-BPG goes on to become 2PG. That leaves 5% of it that can escape the enzyme. So a small quantity escapes the enzyme. If I have a lot of glycolysis going on, I will have more 2,3-BPG released. Therefore, the more 2,3-BPG is released, the more glycolysis is going on, and therefore the cell is giving the signal, hey, I need oxygen. Okay? It's heat, isn't it? It's the heat. Yes? Is more intermediate made. Certainly, the more of this we start, the more of this there will be. So as cells need energy, what do? They burn glucose. The more glucose they burn, the more 2,3-BPG they will produce. Right? So 2,3-BPG is a measure of how much glycolysis is going on in a cell. Yes? No. A new phosphate. Good question. New phosphate being attached. As an intermediate. So you have 2 phosphates on there as an intermediate. So one of them, so you start with number 3, you get 2,3-BPG, then all you have to do is clip off number 3 to be left behind with number 2. Right? Okay. So that enzyme is responsible for producing 2,3-BPG, that is the indicator molecule for hemoglobin. Okay. Well, the next steps are pretty straightforward. 2-phosphoglycerate is converted to PEP. PEP has a longer name. It's called phosphoenol pyruvate. You don't need to know that. You can call it PEP, as most people do. Okay? That's catalyzed by the enzyme known as enolase, and that is simply a loss of water. Water is being lost in that process. Okay? The last step is this reaction here, catalyzed by pyruvate kinase, and this is the reaction I call the Big Bang. The delta G 0' for this reaction is one that I showed you earlier in terms of the high energy molecules. The delta G 0' for this reaction is about minus 32 kilojoules per mole. That's about as much energy as there is in an ATP. Now, what that means is that this reaction is releasing a lot of energy, and this reaction also produces ATP. So in addition to producing ATP, it's releasing enough energy to make another one. There's actually enough energy in this reaction to make 2 ATPs. A lot of it is being lost as heat. Now, if you wonder why you get hot when you go running, this is one of the reasons why you get hot when you go running. The more of this pathway you run, which is what your muscles are doing to make energy, the more of all these reactions are running, the more of this very energetic heat reaction is running, the more hot you are going to get. Okay? So the Big Bang has some significance there. Because this reaction is making ATP, we're now finally ahead on ATP. 2 ATPs up here, 2 ATPs down here. 2 went in, so we have a net product of 2 molecules of ATP. Yes, Maria? First 2 ATPs come right here. Okay? This reaction produces ATP. That was the substrate level phosphorylation. This is another substrate level phosphorylation. Okay? So now we can summarize what's happened in glycolysis. We started with 1 molecule of glucose. What did we end up with? We ended up with 2 molecules of NADH. We ended up with a net of 2 molecules of ATP. And we ended up with a net of 2 molecules of pyruvate. Pyruvate has a lot of energy still left in it. That energy will be realized mostly in the citric acid cycle. We'll talk about that next. Well, actually, after we finish carbohydrates, that is. Okay, So that's what glycolysis is producing. Everybody understand that? Okay. Well, let's turn our attention then to regulation. Regulation turns out to be really important for metabolic pathways because cells don't want to just be all the time burning up all their glucose, because if they did, they wouldn't be around for very long. Glucose is an energy source. It's needed to keep cells alive, and cells have a sort of a basal need for that. But as we will see, cells sometimes don't want to be running glycolysis, and some cells in our body want to be making glucose. We'll talk about that in a bit. But for right now, let's talk about the regulation of glycolysis. Glycolysis reactions look like this, as you can see. Here's that splitting where we got one of those, and then this got converted back into that, so we then had two of all these guys down here. The enzymes that are regulated in glycolysis are the following enzymes, and you need to know this. Hexokinase, that's the first one. It's not unusual that that a pathway is regulated by its first enzyme. Okay? It's an unusual regulation. I won't go into it too much. It's controlled by actually its product. So it's an unusual regulation. Glucose 6-phosphate inhibits the enzyme. The most important enzyme for regulation is actually this one right here, PFK. Phosphofructokinase is the most important enzyme regulating glycolysis. Now, this is unusual. Having a pathway regulated by an enzyme internal to it isn't very common. Let's look to see what regulates this enzyme. It's inhibited by ATP. What's ATP an indication of in a cell? High energy, right? High purines, yeah, but okay, you said high purines. It is an indication of high purines, but more importantly, it's an indication of high energy, since ATP is the gasoline of the cell, right? Measure of high energy. Okay, so it's inhibited by that. This makes sense. Do we want to be burning glucose when the cell has plenty of energy? No. We don't want to be lighting our fireplace in July, do we? Unless we live in the southern hemisphere, right? Okay. If we're sitting up here in July and it's summertime, we don't want to be burning our fireplace because we're wasting wood and we're producing heat that's of no use to us other than making us hot, right? So we don't want to be running glycolysis when the cell has plenty of energy. ATP is a measure of energy. When do we want to run glycolysis? We want to run glycolysis when the cell has no energy. And guess what? AMP is a measure of no energy. AMP concentrations go up when cells run out of energy. Now, this other molecule, F26BP, I'm going to talk about when I talk about gluconeogenesis. So just kind of keep that one stored in your head. But you'll notice it's activated by F26BP. So PFK, a lot of numbers and letters there, is activated by F26BP. And notice 26BP is not the same as 16BP. Okay? All right. Now, this reaction is limited by the amount of NAD that's available. And the reason for that is that this is the only oxidation reaction. This is the glyceraldehyde 3-phosphate dehydrogenase, the G3PDH reaction. This one requires NAD because it's got to have a place to put those electrons that are produced by the oxidation. So consequently, this reaction requires NAD. For all of you who are into fermentation science, or even people who aren't like myself who like to drink beer, this reaction is why brewing is even possible. We'll see that in just a little bit. Tessa? This is the step where the first oxidation occurs. This is where the first ATP is made. Okay? So remember, the oxidation is necessary to make the 1,3-BPG that is then used to make the ATP in the next reaction. This reaction or this reaction? The one that's being limited by NAD is G3PDH. Glyceraldehyde 3-phosphate dehydrogenase. Okay, now, the third enzyme, these three enzymes are all regulated allosterically. Okay? Binding of a small molecule affects the enzyme. This one, this one, this one. This one's simply limited by substrate. The substrate being NAD, which is used to carry the electrons. So the third enzyme that's regulated in the pathway is pyruvate kinase. That's the Big Bang enzyme. Now, why in the world is the last enzyme of the reaction allosterically regulated? Why does the cell want to regulate the last reaction instead of just regulating the earlier ones? Well, the answer to that question is due to the way that gluconeogenesis works. We will see that gluconeogenesis goes backwards for a lot of these reactions. But it uses different enzymes for this reaction, and this reaction. The three allosterically regulated enzymes have different enzymes in gluconeogenesis. All of the other enzymes in gluconeogenesis are the same as the ones in glycolysis. They simply run the reaction backwards. Okay? They simply run the reaction backwards. So why am I saying this about this guy right here? Well, let's look first of all. This enzyme is inhibited by ATP. Same reason as before. We have high ATP. It means we have high energy. We don't want this pathway going, so we turn this guy off. We turn this guy on with AMP, low energy, and we turn it on with something really interesting, F16BP. Now, this is something I alluded to earlier that I want to bring fully to the front right now, okay? F16BP is this guy right here. Look at this guy right here. An intermediate in the pathway is activating an enzyme late in the pathway. Why in the world? Well, let's imagine that you are a runner or you're someone who's been scared and your body says, got to get out of here, got to run fast. Muscles start contracting to run or to push off, exercise, whatever it is. And what happens? Your glucose starts getting burned, and it goes to glucose 6-phosphate, glucose 6-phosphate to fructose 6-phosphate, fructose 6-phosphate to fructose 1,6-bisphosphate. And if you remember what I said about the aldolase enzyme, I told you that it had a delta G0 prime that had a very positive value. I said it was an energy hump that the cell had to get over. An energy hump. Well, what does an energy hump mean? It means it's not a real favorable reaction, right? And how does a cell overcome an unfavorable reaction? By manipulating concentrations of products and reactants. What happens when this reaction is blocked? This is going to accumulate, right? That is a reactant for the aldolase enzyme. We're starting to make reactant. More reactant means that it's going into the denominator, meaning that ratio gets smaller, meaning that the log term gets more negative, and we start favoring the reverse reaction. However, there's another component to this. The other component is, as this concentration goes up, it starts affecting this enzyme down at the bottom. What does that do? Well, if I turn on this enzyme, what's going to happen to this reaction? It's going to go forwards, right? And it's a very energetic reaction, so it's going to go strongly forwards. What's that going to do to this reaction? When this product starts disappearing, this will start going forwards. And this starts disappearing, this will start going forwards. And this starts disappearing, this will start going forwards. And what we see is a chain reaction of pulling all of these reactions forwards. And that has the effect of decreasing the concentration of these two. So look what we've done. We've increased the concentration of this one, And by this one activating this enzyme, we've decreased the concentration of these two. Consequently, the aldolase reaction becomes very favorable. And the cell can get the energy it needs as a consequence of that. And that's really cool. This is a phenomenon I call pushing and pulling a metabolic reaction. I'm going to define those for you. Pushing a reaction. What does pushing a reaction mean? It means that you start increasing the amount of reactant. Pushing it. Right? What does pulling a reaction mean? Pulling means you start decreasing the amount of product. And here's the product. So through the combination of pushing and pulling, the aldolase reaction becomes very favorable. It's a cool trick that the cell is going through to make sure that this pathway is running when it needs stuff. Questions about that? Yes?
So you mentioned the Big Bang reaction creates excess heat.
Yep.
Does that help that reaction go forward?
Heat doesn't have any big impact here. The amount of heat that's released is not significant enough to have a big impact on the reaction. Heat does affect the Gibbs free energy equation because it's the delta G0 prime plus RT, that's the temperature, times the natural log of the products over reactants. But the temperature in a cell like in our body doesn't change significantly as a consequence. But yes, it has a very minor effect. Okay, other questions? Cool process. All right, let's talk about a couple of things then. Something of interest to the brewers. All right, so I told you that the G3PDH requires NAD. It's got to have NAD+. And NAD+ is needed because that's an oxidation reaction. We've got to have something to put electrons on. Normally, if oxygen is present, NADH is converted to NAD by the electron transport system. We'll talk about that soon. Whenever oxygen is present, NADH is converted back to NAD. That gives NAD for the G3PDH reaction, and everybody's happy. However, when cells run out of when cells run out of oxygen, the electron transport system will not run. Consequently, NADH will not be converted to NAD. And unless something is done, the glyceraldehyde 3-phosphate dehydrogenase reaction cannot run. Okay? Let's take a look at that real quickly. Okay? Okay. Here's the glyceraldehyde 3-phosphate dehydrogenase reaction, and you see that NAD is needed for that reaction to go. This was the only oxidation in glycolysis. Okay? If the cell doesn't have a way of regenerating NAD, this reaction is going to stop. The cell's going to die. It won't be able to produce energy that it needs, especially when it needs it, because when it's running out of oxygen, it's usually because, in the case of a muscle cell, it's exercising pretty heavily. Well, to take care of that situation, at least over the short term, cells have an alternate way of making NAD. They have an alternate way of making NAD. The alternate way that cells have of making NAD when there's no oxygen is known as fermentation. Fermentation exists solely to make NAD to keep glycolysis going. It's the only reason fermentation exists. Now, here shows the reaction that actually occurs in a bacterial or yeast cell. What do? They take a compound called acetaldehyde, which is made from pyruvate. Remember, pyruvate's an end product of glycolysis. Pyruvate gets converted into acetaldehyde, and then acetaldehyde gets converted into ethanol. And it's done using electrons from NADH that produces NAD. And consequently, the cell can keep this thing going. The beauty for all of us is it's continually producing ethanol. Okay? Everybody's happy. The reason that when you are brewing beer, you cap it off so that with a small volume in the top of the thing is so that you limit how much oxygen that they have. And when they don't, and then when they run out of oxygen, they start running this reaction. This enzyme has a name of alcohol dehydrogenase. Okay? Now, I want you to remember that for just a second. And I want you to remember what reaction. I'll come back to this slide in just a minute to show you how this is made. Notice this says alcoholic fermentation, implying that there are other kinds of fermentation, and there are. One of the kinds of fermentation you have in your body. We all have in our body. And it's the first reaction that I showed you. Pyruvate goes to lactate. The same thing is going on. Look, NADH is producing NAD. Pyruvate is getting converted as a consequence into lactate. Notice we have a ketone here. We have an alcohol over here. Lactic acid is produced when our muscles run out of oxygen. And yes, they do run out of oxygen. When we exercise very heavily, we can be using oxygen faster than our blood supply can deliver it. Okay? That means that the cells don't have a backup like this. The muscle cell is going to die pretty quickly. So muscle cells ferment in order to produce NAD, just like bacterial and yeast cells do. They just use a different reaction. Yes, sir? Yes.
When oxygen returns to the system, does the ethanol or lactic acid get?
Very good question. So if I am running out of oxygen and my muscle cells start producing lactate, if I get oxygen back, will this reaction run backwards? In fact, it will. And there's a very cool process that occurs in our body called the Quarry Cycle that explains that. But let me save that until we get to the Quarry Cycle, because this turns out to be significant in the metabolism of our body. Okay? The efficiency? What do you mean by efficiency? I'm not sure I understand.
I mean, like, what, like at the end of the cycle, just producing one more thing of NAD.
So you get one NAD every time you run this reaction. Right, right. Okay. Now I'll tell you something surprising. Let's go back to the last slide. The surprising thing is right here. You have this enzyme in your body. You just can't do this reaction. Why in the world do you suppose you have that enzyme in your body? If you can't make this from pyruvate, what use is this enzyme? It'd be really nice to not have to buy beer on Friday night, right? Or Thursday night, as the case may be. So you can run the reaction backwards. And what do you do when you run the reaction backwards? You get hung over. Your body's trying to get rid of too much ethanol, and that's the reason that you have that enzyme. It converts it to this. This can actually get converted to acetic acid, both of which, acetic acid and acetaldehyde, can contribute to a hangover. Yes? If your body didn't produce that enzyme, you probably would have toxic amounts of ethanol. You know about people who have ethanol toxicity from drinking too much? What they're doing is they're overloading that enzyme. So the amount of ethanol you would be able to tolerate is probably fairly small if you didn't have that enzyme. You may know people who, people who have ethanol sensitivities, for example. A little bit of ethanol makes them very sick. It's because they don't have much of that enzyme either. Yeah. Yes, sir?
So, if I drink all the time, and my sensitivity is higher, does that mean I'm going to have more alcohol?
Yes, the question is, if I drink more and more and more, my sensitivity to ethanol goes down, means I can drink more and have less effect, right? Does that mean that I am making more of this enzyme? Yes. Yes. So the alcoholic over time is making a lot of this enzyme. A lot of it, a lot of this enzyme. Yep. Yes? Is this contained in the pill they give to alcoholics to keep them from drinking? I don't think so, but I don't know enough about that pill. I know it makes them ill, but I don't think it's this compound, no. Okay, so that's what's up with fermentation. We do fermentation to make NAD. There's the pyruvate. By the way, this reaction is catalyzed by an enzyme we call pyruvate decarboxylase. We'll talk more about that enzyme later. Don't worry about that enzyme name right now. Important vitamins in the metabolism of pyruvate, thiamine. Okay, thiamine's converted into this guy here, thiamine pyrophosphate. Okay, that should be TPP, not TTP. TPP. Thiamine's converted into that, and this is necessary for the metabolism of pyruvate, as we will see soon. And finally, lactic acid fermentation. There's now lactic acid fermentation showing exactly the same thing that we saw before with alcohol dehydrogenase, the product here being lactate. Some people think that production of lactic acid is why your muscles get sore when you exercise heavily, that you're producing this guy as an acid that's a fairly strong acid, and it may be damaging to muscle tissue. Other people dispute that, so it's not completely clear. But one thought is that lactic acid is, in fact, the cause of muscle soreness. Okay, that's where I want to stop for glycolysis. I do want to talk about gluconeogenesis in preparation for your exam, though. Gluconeogenesis right here is where I'm going to stop for the exam. So let's talk about gluconeogenesis. Gluconeogenesis is, in a lot of ways, the reversal of glycolysis. Glycolysis is the breakdown of glucose to make pyruvate. Gluconeogenesis is the use of pyruvate to make glucose. Okay? The use of pyruvate to make glucose. Here's a scheme of the overall pathway. Going down, we're looking at glycolysis. Going up, we're going through gluconeogenesis. And what I've done is I've highlighted the reactions that are different in gluconeogenesis compared to glycolysis. It turns out that 3 of the reactions in glycolysis are replaced by 4 reactions in gluconeogenesis. Okay? 4 reactions require 4 different enzymes to do gluconeogenesis compared to glycolysis. All the other enzymes are the same. It's just simply a reversal. So 7 enzymes of glycolysis match 7 enzymes in gluconeogenesis. It means, therefore, that gluconeogenesis has 11 enzymes, whereas glycolysis only had 10. So what I want to do in talking about this pathway are talk simply about the things that are different. That is, the enzymes that are different. I'll start here at the bottom, and we'll move upwards. Let's do that. Let's start with the first reaction. The first reaction of gluconeogenesis is the conversion of pyruvate to oxaloacetate. This is a reaction that requires ATP and it requires carbon dioxide. The reason it requires carbon dioxide is because we're going to make a 4-carbon intermediate right here, oxaloacetate. Yes, you should know the names and enzymes here too. Okay? You're going to make oxaloacetate that has 4 carbons. That requires energy from ATP. It requires the enzyme known as pyruvate carboxylase. Now, what's all this other stuff on here? Well, this is an activator of the enzyme. We're not going to worry about that at the moment. This, however, is a vitamin. And this vitamin is needed by every carboxylase because what it does is it carries the CO2. Biotin carries the CO2. It's a coenzyme for the reaction. So oxaloacetate appears in gluconeogenesis. It doesn't appear in glycolysis. Okay? It's the only molecule that doesn't appear in glycolysis. Yes, sir? Okay, biotin is both a coenzyme and a vitamin. Yes. Okay. We don't need that. Let's forget that for the moment. Don't want to confuse things. Right? There's biotin. And biotin carries the carbon dioxide out here. Okay? You don't need to worry about that, but I'm just showing you what it looks like. Biotin is actually covalently bound to the enzyme. Let's look at the second reaction of gluconeogenesis. Remember, starting at the bottom and moving upwards. The second reaction converts oxaloacetate into PEP. Now, notice what we had to do in gluconeogenesis. We had to use two reactions to go from pyruvate to PEP. That's not totally surprising because if you remember, I said that the Big Bang reaction produced an ATP and released enough energy equivalent to an ATP. That means there's a lot of energy, a big barrier we've got to get over to get from pyruvate back to this guy. Okay? So it's not surprising that the cell is doing it in two steps. The first step was making oxaloacetate. The second step is making PEP. Look at what we got here. Another molecule of energy required to make this. In this case, we're using GTP energy in order to produce PEP. This enzyme, long name, you can call it PEPCK. PEPCK. This enzyme is not made by all cells of the body. It's only made in your liver and in your kidney. And it turns out that's the only tissues in your body that can do the synthesis of glucose. The only cells that can do the synthesis of glucose, liver and kidney, parts of the kidney. It's not even the whole kidney, just parts of the kidney. And it's because this enzyme isn't made in most cells. That's our third type of regulation we talked about. Remember allosteric regulation? Remember covalent modification? And the third I said was whether or not an enzyme was made. This is an example of an enzyme that's controlled by whether or not it's being made. Did you have a question back in the back? Oh, I thought you had your hand up. Okay. You guys with me? We're almost there. We're almost there. The third enzyme that's different. Remember, all the other reactions are just going backwards until we get all the way up to fructose 1,6-bisphosphate. PFK is not used in gluconeogenesis, just like pyruvate kinase is not used in gluconeogenesis, right? We use two enzymes to go around it. PFK is not used. Instead, a different reaction is run. We don't reverse the PFK reaction. Instead, we do a different reaction. Here's the reaction that we do. This guy loses a phosphate. If we were to reverse the PFK reaction, we would have to be putting that phosphate back onto ADP to make ATP. We don't do that. We simply clip a phosphate off and produce fructose 6-phosphate. The enzyme? F16BPase. F16BPase. That's the enzyme that replaces PFK. I'm sorry? This makes it, it's a good question, this makes the reaction energetically favorable. If we had to try to remake that ATP, it would be energetically unfavorable. Okay, We're almost there. There's one more reaction that we have to bypass, and that's the hexokinase reaction. And that's bypassed in a manner that's almost identical to the last one. We clip a phosphate off instead of regenerating ATP. This reaction is catalyzed by G6Pase, glucose 6-phosphatase, G6Pase. This reaction occurs in the endoplasmic reticulum. All the reactions of glycolysis occur in the cytoplasm. There's only 2 reactions in gluconeogenesis that occur out of the cytoplasm. The first one and the last one. This is the last one. It occurs in the endoplasmic reticulum. The first one was the pyruvate carboxylase reaction. It occurred in the mitochondrion. It occurred in the mitochondrion. So pyruvate carboxylase is in the mitochondrion. Glucose 6-phosphatase is in the endoplasmic reticulum. And all the other enzymes are in the cytoplasm. I told you I'd tell you about F26BP. Here it is. It sounds like it's an intermediate in glycolysis. It's not. F16BP is, F26BP is not. Don't worry about how it's made. Everybody gets all worried about how did you make it. An enzyme catalyzes its formation. Does that tell you anything? Okay, don't worry about it. It's not made in glycolysis. It's made elsewhere. This molecule, however, turns out to regulate glycolysis very well. This molecule is responsible for a phenomenon we call reciprocal regulation. It's a very important principle in controlling metabolic pathways. Reciprocal regulation. You'll see why we call it reciprocal regulation in a minute. Remember the PFK reaction? The PFK reaction went in glycolysis and was replaced in gluconeogenesis by F16BPase. PFK going down, F16BPase going up. This guy being gluconeogenesis, this guy being glycolysis. F26BP activates PFK. It inhibits F16BPase. Has opposite effects on catabolic and anabolic pathways. That's what reciprocal regulation is. One molecule having opposite effects on catabolic and anabolic pathways. All right? So it activates the PFK. It inhibits the F16BPase, the corresponding enzyme in gluconeogenesis. Very simple. The cell makes this one molecule. What's going to happen? The cell is going to do glycolysis. It's not going to do gluconeogenesis. If the cell stops making this molecule or breaks this molecule down, it's going to stop running glycolysis, and it's going to start running gluconeogenesis. Reciprocal regulation. Very simple, very, very easy for it to control. The last thing I'm going to tell you is the easiest thing I'm going to tell you all day. I know you've had a lot of stuff here. The Quarry Cycle. The Quarry Cycle is a very cool cycle in our body. It operates between our liver and our muscles. Remember that our liver, I said, was one of the 2 tissues that makes glucose in our body. Muscles use glucose, but they can't make it. Your eyes use glucose. They can't make it. The Quarry Cycle operates between your muscles and your liver. Let's imagine I'm going out on my morning run, which I'll be doing in the morning. Anybody wants to join me? Okay. In the morning, I'll be running. And what's going to happen in the morning is I'm going to take off running, and my muscles are going to start using glucose because they want to make ATP energy. They start breaking down my glucose. And I hate to say this, but even an old guy like me can run fast enough that I can outrun my body's ability to supply oxygen to my muscle tissues. why I slow down after a while, right? What happens when my muscles start running out of oxygen? What do? What process? Fermentation, right? I do fermentation so I can make NAD to keep glycolysis going in my muscle cells. What's the product of fermentation in my muscle cells? Lactic acid. Lactic acid is of exactly zero use to my muscle cells. So the muscle cell says, get out of here. It dumps it into my bloodstream. So the bloodstream carries away the lactic acid from my muscles. And where does it go? It goes to my liver. And you might say, well, why doesn't your liver run out of oxygen? And I say, my liver is right next to my lungs. My liver's got plenty of oxygen. What does my liver do? It takes that lactate reaction backwards and makes pyruvate. And since the liver has plenty of oxygen, it's got plenty of ATP too. So what does the liver do with that pyruvate? It makes glucose. And the liver says, I don't need all this glucose. What am I going to do with it? Oh, I'll dump it in the bloodstream. And it goes back to the muscles. So the Quarry cycle is what I just described to you. Muscles produce lactate. Liver uses the lactate to make glucose, puts it back in the bloodstream for the muscles. And it just goes on and on and on. My liver is supplying my muscle cells with glucose. Really, really cool, really, really critical. Okay? That makes sense? Yes, sir.
As far as like lactic acid, what I was always told, as far as like, I was a pitcher, so whenever we pitched, afterwards, they really had a run after every game. The lactic acid built up in your shoulder.
Uh-huh.
And if you start running too much, then aren't you just getting more lactic acid buildup? Because the point was to get the lactic acid into the bloodstream quicker, right?
Right. That sounds to me like it's not biochemically sound.
That's what I'm hearing right now.
Okay? So there's a lot of urban legends out there about a variety of things. That's not biochemically based. Right. Pitching is much more likely to involve mechanical problems with your arm than it is to involve things relating to lactic acid. The reason being that pitching itself is a short-lived phenomenon. That act of doing the pitch doesn't take that long to have that happen. So I think that's baloney, to be honest with you. Tell your coach. Okay, questions about that? Pretty cool stuff. Now, one thing I want to mention, actually, this is one last thing to remember. This is kind of cool. All right? This is going to surprise you. When cells are doing fermentation, okay, the energy they're getting from glucose is 2 net ATPs per glucose, because the NADHs don't count. They get recycled, right? So in the absence of oxygen, all that cells are getting in fermentation is 2 ATPs per glucose. If oxygen is present, depending on how we count it, and I tend to count on the high side, but if oxygen is present, each glucose gives 38 ATPs. That's 19 times as much ATP per glucose when oxygen is present compared to when it's absent. What's that? 38 net ATPs. My question to you is, thinking about all the things you're told about exercise, right? You decide you're going to go on the diet, you're going to start exercising, you're going to go out, and I want to go to the gym, and I'm going to get what when I start moving around? Aerobic? Do you really want to get aerobic when you want to lose weight, or do you want to get anaerobic? Which one's going to burn more ATP? Or more glucose to make ATP? When people talk about going aerobic, they're usually talking about going anaerobic. Okay? That's why you start breathing heavily. Because you're going anaerobic, your body's trying to make up the deficit. But they have this notion that they're going aerobic. They're actually going anaerobic. You lose the most weight when you go anaerobic, not when you go aerobic, folks. Tell your friends that too. That's another urban legend. Where am I? It would be right here. It's the supercalifragilisticexpialidocious, if you remember that song. It's a lot of energy, kind of like ATP. When cells have lots of ATP and NADH2, they strive to store this energy as sugar. Yes, they do. Inside of mitochondria, they start with pyruvate, carboxylating it to make oxaloacetate. Oh, gluconeogenesis is so exhilarating. Memorizing it can really be exasperating. Liver cells require it, so there's no need for debating. Gluconeogenesis is so exhilarating. Glucose, glucose, come to be. Glucose, glucose, come to be. Oxaloacetate has got to turn to PEP, employing energy that comes from breaking GTP. From there it goes to make a couple phosphoglycerates, exploiting enolase and mutase catalytic traits. Oh, gluconeogenesis is liver specialty, producing sugar for the body most admirably. 6 ATPs per glucose is the needed energy. Gluconeogenesis is liver specialty. Oh, glucose, glucose, joy to me. Glucose, glucose, joy to me. Converting phosphoglycerate to 1,3-BPG requires a phosphate that includes ATP energy. Reduction with electrons gives us all an NAD, and G3Ps isomerize to make DHAP. Oh, gluconeogenesis is anabolic bliss, reversing 7 mechanisms of glycolysis. To do well on the final, students have to learn all this. Gluconeogenesis is anabolic bliss. Oh, glucose, glucose factory. Glucose, glucose factory. The aldolase reaction puts together pieces so a fructose molecule is made with 2 phosphates in tow, and one of these gets cleaved off by a fructose phosphatase unless F26BP's acting blocking pathways. Oh, gluconeogenesis, a pathway to revere that makes a ton of glucose when it kicks into high gear. The cell's a masterminding metabolic engineer. Gluconeogenesis, a pathway to revere. Oh, glucose, glucose, jubilee. Glucose, glucose, jubilee. From F6P to G6P, it is the final phase. Oh, I've lost my thing here. Come on, guys. The enzyme catalyzing it is an isomerase. Then G6P drops phosphate and a glucose it becomes inside the tiny endoplasmic reticulums. Oh, gluconeogenesis is not so very hard. I know that on the final we will not be caught off guard because our professor lets us use a filled out index card. Gluconeogenesis is not so